as I've shown here, or also through changes in resource quality, especially when you're thinking about plant-insect interactions. An early arriver can really alter the quality of a shared resource through things like induced defenses. And so what's interesting about priority effects is that it's kind of this idea that this seemingly stochastic or random order of arrival can dictate the outcome of community assembly, which is not very satisfying from a scientific perspective, where we're trying to understand kind of common rules and kind of generalities. So this idea that stochasticity can play a huge role. So a lot of research in on priority effects has focused on understanding when these kind of alternative stable states or priority effects are most likely to occur. So we can try to kind of impose some kind of framework into understanding and getting more general and scientific about it. And so I was interested in a kind of abiotic pressure that I thought would alter the likelihood and strength of priority effects. And I thought this because the timing of arrival should matter when early arrivers have a strong impact on a shared resource. And so this occurs when early arrivers can have fast growth rates and rapid resource depletion of this shared resource. And we know that temperature tends to increase biological rates like growth and feeding up to some critical point where it declines for ectotherms. We've known this for a long time, but more recent extensions of metabolic theory have also shown that this effect of temperature on these rates is really different across different taxa. And so because respiration responds more quickly than photosynthesis to temperature, what this means is that as temperatures increase, consumers start to outpace their resources. And so this should lead to faster growth rates and not only faster growth rates, but also faster resource depletion as the consumers start to kind of overtake their resources, and thus a stronger impact of the early arriver on this shared resource. So for this project, I was predicting that warming would speed up resource depletion, which would make arriving first more important. And also, as kind of a side effect, increase dispersal rates away from these kind of overtaxed and declining resources. And I was interested in looking at dispersal because, of course, dispersal is what will ultimately dictate which species will arrive first. So the system that I used for this project were the aphids that compete on common milkweed. And so milkweed is this highly defended plant that has these toxic carnitinoids and latex and trichomes that defend it. And so only a very small group of insects can actually eat it. And the ones that eat it are really specialized on milkweed. And that includes two species of aphid, Aphis nerei and Aphis asclepiatus. And for this talk, because I'm going to jump through so many different systems, I'll just call these the yellow aphid and the green aphid for simplicity. And what we know about them is that they both induce milkweed defenses. But overall, the yellow aphid is just better at most things. So it's a better competitor, better disperser. It's also better at kind of tolerating predators. So it tends to outcompete the green species. But it's not able to lay eggs, so it's obligately parthenogenetic, whereas the green species does lay eggs. And that means that in Southern Ontario, where I did this study, it's able to overwinter. So it overwinters as eggs, and each season it's able to arrive first. So it emerges from these eggs and arrives first, and the yellow species actually gets blown up from southern populations. So what this means is that the milkweed kind of opens in the spring, the green species arrives first, and then the yellow arrives later, and then they compete throughout the rest of the season. And so this early arrival of the green species has kind of been hypothesized as one way that it might have an advantage over the yellow species. So this kind of priority effect. So in the experiment I conducted, I looked at, I had a treatment that was kind of this natural scenario with the green species first, scenario with the yellow species first, both species arriving at the same time, and then plants that had no aphids on them. And then I crossed this with a warming treatment. And I looked at aphid population dynamics, and today I'm going to focus on population extinctions, plant quality. So I looked at a few different defenses, and today I'm going to talk about trichomes. And from the aphid perspective, more trichomes is lower quality. Plant quantity, and I'll focus today on mortality. So when they die more, there's lower quantity of plants available to them. And then I put sticky cards at the end of the cages that the plants were in to track aphid dispersal throughout the experiment. And so I'll talk about that as well. So to remind you, I predicted that warming would first speed up resource depletion. So here I'm showing these two plant responses, trichome density and milkweed mortality, as a function of aphid abundance, because both are kind of affected by how many aphids are on the plants. And I'll show the unwarmed in blue always and warmed in red. And so in the plants that had no aphids, there was no difference between, there's no effect of warming, no difference between the unwarmed and warmed plants. But then for trichomes, as aphid abundance increased, so did trichome density. And the per capita kind of impact of an aphid on trichome density was higher under warm temperatures. So we get more trichomes with warming. And likewise, we see that the aphids are inducing mortality in the milkweed, and we get this more so in the warmed conditions. So warming reduced milkweed quantity and quality. And we predicted that this would make arrival order more important. And so now I'm showing the population persistence of these aphid populations when they arrived before their competitor, at the same time as their competitor, or after their competitor. And again, I'll show unwarmed in blue and warmed in red. So it's not very interesting for the yellow species because, as I mentioned, it's this kind of superior competitor and none of its populations went extinct. But the green species didn't do as well. And so we see that arriving late is not great for the population persistence, and warming is not great for population persistence. And we get this kind of interaction between the two, where the effect of warming on decreasing population persistence is much stronger under warmed conditions. So they go from about 90% populations surviving down to 10 under the warmed conditions. So we see that kind of warming made arriving early more important. And then we predicted finally that warming would increase dispersal away from these dying plants. And now I'm showing per capita dispersal over the course of this 5-week experiment. And for the yellow species, we get pretty much what we predicted, where we see that dispersal tends to increase over time as the plants die, and this occurs more rapidly in warmed conditions. However, for the green species, we don't see it. This was no effect of temperature. And if anything, they're dispersing less under warm conditions, and they disperse less overall. So warming actually has strengthened these differences in dispersal between these two species. So to summarize this, warming for the resource strengthened the declines in the plant quantity and quality. It made arriving early more important for this weaker competitor. But it also potentially made it less likely that the weaker competitor would arrive first, because it increased dispersal rates only for the stronger competitor. So what does this mean? Well, I tried to do this as kind of a first link between metabolic theory and the known effects of temperature on trophic interactions, linking that to what we know about the impact of arrival order, and as well as dispersal. This hopefully tells us something about global change responses in these systems, which there are a lot of, that are temporally and spatially complex, because this kind of integrates this temporal aspect as well as this spatial aspect, and kind of indicates that climate change can make timing more important. And so I think the next steps for priority effects research is kind of understanding how widespread they are in nature, and continuing to work on what makes them likely to occur, because I think there can be kind of a perception that may or may not be accurate, that this is like a very specific dynamic with these priority effects, that occur in specific systems. And I still need to be convinced that this is a general phenomenon that can occur across systems. And so documenting that, working with a group, an IDIV working group that's kind of trying to present these kind of general findings on priority effects. So the next project also has to do with priority effects, but from a different kind of theoretical framework. And so I did this experiment at Stanford, working with Tad Fukami and my PhD advisor, Ben Gilbert, and Andrew Ludden. So this project comes from a different perspective, which is thinking about how competing species coexist. And so coexistence is maintains biodiversity, and so people are very interested in understanding how it works. And when two species coexist, it requires some kind of mechanism that reduces competition between these competitors, and creates a scenario in which each species does best when it's rare and its competitors common. And so what this creates is a situation in which each species can invade a established population of its competitor. And so here we have blue invading green, and we have the invader population size going up over time. And likewise, with these coexisting species, we get green is also able to invade blue. And so Chesson's modern coexistence theory, among other things, introduced the idea that there are two types of species differences that determine whether or not species can coexist. The first is probably the more familiar, which is these niche differences or stabilizing differences, which are differences in things like resource use, predators, or pathogens that reduce both species' sensitivity to interspecific competition and promote coexistence. And so they allow species to invade from rare. And what is maybe less familiar to people outside this framework are these ecological fitness differences. And so this is differences in things like growth rate and sensitivity to competition that simply make one species a better competitor. And they promote competitive exclusion. And so we end up with this kind of result where the outcome of competition, whether or not species are able to coexist, depends on the fitness differences and their stabilizing differences. And so here, the green and blue species I showed before are able to coexist because they have strong enough stabilizing differences and low enough fitness differences. But now, say we had a species where there was more kind of niche overlap between these two. And here, so we have lower stabilizing differences and the same fitness differences. And now yellow cannot invade green. And so green outcompetes yellow and we get competitive exclusion. So when I conducted this study, there was already like a rapid growth in the use of modern coexistence theory. And people tend to use it to understand how some factor will impact the outcome of competition and coexistence outcomes. And some classic examples are looking, so this 2015 paper looking at the effect of kind of traits and how traits relate to coexistence outcomes, and this 2013 paper looking at the effect of phylogenetic relatedness on coexistence. And so the results are laid out like this, with fitness differences along the Y, stabilizing differences along the X. And I'll draw your attention to the 0 point, because you can see here that some points fall below 0, and a lot of points here would have fallen below 0, but they're kind of lined up there. And so this was kind of how the studies were done at the time, where we tended to focus on these two outcomes, competitive exclusion and coexistence, and ignore any points that fall in the negative. But there's another side of coexistence theory, and that's what my project was focused on. So I've talked about cases on this side where individuals are limited by members of their own species and you have an advantage when rare. And this kind of makes intuitive sense from a classic like niche partitioning kind of framework. But you can also get a case where individuals are limited by members of other species and you actually get an advantage from being common. And this had been pointed out in the literature at the time. And so you might think, how does this happen? Well, if you're thinking about niche differences, it's kind of difficult to envision a negative niche difference. So that's why we call them destabilizing or negative stabilizing differences. But you can think about scenarios in which an organism might have an advantage from being common, like allelopathic organisms that release chemicals that are toxic to other species, or even pollinator dynamics where you might get benefit from being around members of your own species that are also attracting pollinators that you need. And so in these cases, we had both species invading for coexistence, one species invading for competitive exclusion. And now we get a scenario where neither species can invade an established population of its competitor, and whichever species arrives first wins. Well, hopefully that sounds familiar, because I just talked about a whole field of research that is focused on the idea that the order of arrival matters for the outcome of competition. And so that's called priority effects. So what's interesting is at the time, there's a field of priority effects looking at the importance of arriving first. This mathematical framework of coexistence theory has this very nice kind of way to integrate priority effects, but it was completely ignored, the outcomes, partly because these outcomes of priority effects were, I think, were perceived to be not very common in these competitive experiments. So in this project, I first set out to ask, can we use modern coexistence theory to study priority effects? And what this requires is species pairs falling in this zone of priority effects. So low enough fitness differences and negative enough stabilizing differences to actually exhibit priority effects. And so I wanted to ask that in a specific experiment, but also across the studies that had been conducted to that point. And so then I wanted to ask what kind of environments would promote priority effects. And so you can promote priority effects by moving species pairs into this zone, either by reducing fitness differences or making stabilizing differences more negative. So I wanted to look at what environments would promote priority effects and how this would occur. And so I did this experiment in nectar microbes, the nectar microbes that live in mimulus flowers. I did this at the end of my PhD in California using 4 Meshnickovia yeast species. And I chose this system for looking at priority effects because this is a system that Tad Fukame has developed there that has been shown for the order of arrival to be really important for the outcome of community assembly. And so for this system, the flower opens, bees and birds pollinate and drop these microbes into the floral nectar. They compete, things happen, and then the flower dies after about a week. And so in the floral nectar, we have amino acids, which is what these yeast are eating, sugars, high levels of sugar, which actually are less of a resource and more of an osmotic stress for these microbes, and then varying levels of pH, which can also be a stress for these organisms. And so I conducted an experiment where I wanted to quantify stabilizing differences and fitness differences. And I took out, I'm not going to go into the math of what exactly I did, but essentially what you want for stabilizing differences is for both of these is to quantify species sensitivity to competition. So because stabilizing differences reduce both species sensitivities to interspecific competition, and fitness differences cause species sensitivities to interspecific competition to diverge. And what this requires is measuring each species' growth rate when alone and its growth rate when invading an established population of each of its competitors. And so you see how much their growth rates are reduced when they're invading, and that's their sensitivity to competition. So for each species pair, so here we have red and blue, you simply measure its growth rate when alone and its growth rate when invading. So this is probably the most straightforward, least math-intensive way of doing modern coexistence theory, and that's why I went with it. And I did this across 6 species pairs and 6 nectar environments. And so you can create these kind of fake flower nectar environments in a 96 wall plate. and I created these low sugar and high sugar treatment, and then 3 levels of pH, and then added 22 amino acids to everything. So for the first question, can modern coexistence theory be used to study priority effects? So all the figures I'm going to show you for this project look like this, where we have fitness differences along the y and stabilizing differences along the x. And if you are paying attention, you'll notice that everything here is negative. So that gives you a hint and a spoiler as to whether or not we found priority effects. So in this study, we found a ton of priority effects, species pairs that were falling in this zone, and also as well a lot of competitive exclusion. So we get these negative stabilizing differences, mostly negative stabilizing differences in this study. And then I looked across studies that have been conducted using modern coexistence theory to this point. So here we have to compare what we found with others, because this is a lot of negative stabilizing differences. So 43% of interactions, and across all studies, we get priority effects in 17% of interactions. So not an insignificant proportion of priority effects across studies, and kind of an argument for why they should be included as a potential outcome. And then to the second question, do some environments promote priority effects? So now I'll actually describe these points. So the colors are the different environments. So low sugar is light and high sugar is dark. And then we go kind of red to blue with the pH. And what I did to kind of investigate whether some environments promote priority effects is I took the distance between each of the points within a treatment. So this is high sugar and low pH treatment, it's dark red. And the point to this dividing line between our 2 outcomes, priority effects and competitive exclusion. And this was the average distance here. So this red line, it was this far, and you see that predominantly priority effects was the most common outcome in that environment. And I did that for all the 6 environments to get this kind of like average outcome. And what you can see is that all the dark lines are on this side in priority effects, and all the light lines representing low sugar are in competitive exclusion. So what we found was that low sugar was associated with competitive exclusion, and this high sugar, kind of more osmotically stressful environment was associated with priority effects. And then we looked at whether this was because fitness differences were changing or niche differences or both. And what we found was that it was actually due to change in fitness differences. So increasing the sugar reduced the fitness differences overall. So what does this mean? Well, it's kind of a general indication that these stressful conditions can make timing more important by increasing species competitive equivalency. And then more broadly, that coexistence theory can and should incorporate this third outcome, this priority effects. And as well, I kind of expanded on this to look at other things that modern coexistence theory can and should incorporate, things like trophic interactions, ecoevolutionary dynamics, range limits. So historically, coexistence theory was really focused on local coexistence, but it could do a lot more than that. Oh, and then since I published a study in 2019, I'd say the field since and even before this study was published, the field has gotten a lot better at incorporating priority effects. So this was already happening. I'm not going to take credit for this. Maybe that study played some role, but this was already starting to happen. Studies now, I'd say, much more commonly have this kind of outcome where we actually are including priority effects, which is great. So the last project I'll talk about starts to integrate evolution and looks at adaptation to competition and climate in fruit flies. And I did this project during my postdoc at Princeton with Jonathan Levine, Paul Schmidt, and Seth Redman. So I've talked about how ecological and evolutionary trajectories occur, and these outcomes fluctuate through time. And historically, we would have thought that these are very different timescales, where with ecology occurring rapidly and evolution occurring on a very different, much longer time scale. But over the past few decades, there's been this widely accepted convergence of timescales where essentially you can get evolutionary dynamics happening as quickly as ecological. And so you get all these examples of rapid evolution. And you can see in the literature examples of things rapidly adapting to climate, so things like warming and drought, and to competition. I'd say less so to competition, but there are also some great examples of variety of organisms being able to adapt within a few generations to competitive interactions. But what I was interested in looking at was kind of the interaction between these two pressures. And in particular, in looking at the consequences of what happens when the pressure that each of these exerts on evolutionary trajectories fluctuates through time. So we get pressure from competition in one time period and from climate in the other. And will these exert kind of the same or opposing selection? And so the system I used for this project was fruit flies, using this model system for evolution, Drosophila melanogaster, and this large-bodied invasive competitor, Zaprionis indianus. So Drosophila melanogaster is naturalized in North America and is relatively more cold tolerant, whereas Zaprionis indianus is recently invaded and more warm adapted. And so I did this study in Pennsylvania, and there Zaprionis dies off first each fall because it can't handle the cold. And so what happens is you get your kind of focal species growing, the population's growing rapidly throughout the summer, and they're faced with this competition for their resource, which is rotting fruit over the course of the summer, and then this very stressful cold fall climate. So this fluctuating selection. And so we were wondering how exposure and adaptation to this large-bodied invasive competitor would impact the species' ability to deal with this stressful fall climate. And so you can imagine a scenario in which it adapts, say, to a larger body size, and larger body size is known to be associated with doing better in cold, and so potentially it does better in the fall. But what we don't actually know, we have so little information about general adaptive responses to competition that maybe we didn't even know we didn't even have a real good expectation of which kind of direction selection would take in response to competition. So maybe it will adapt, it will evolve a smaller body size that's able to develop faster and outcompete this invasive competitor, in which case it could actually be detrimental in the fall. And in terms of fall evolution, it was even more of a black box. So what to expect in terms of the impacts of adapting to a competitor in the summer on evolutionary trajectories in the fall. And so the question was, how does a history of exposure to competition affect subsequent ecological and evolutionary responses to a cold fall? And so the experiment that I conducted had two parts, the summer and the fall. And so in the summer, there were 14 populations evolving without the competitor, so evolving alone and growing and adapting to the summer climate, and then 14 populations evolving with this competitor. And the experiment looked like this, where we had this was an outdoor field experiment with fruit flies, where we had about 220 to 100,000 flies per population. And we fed the flies this fly food, which we were able to manipulate. So they eat the food and then they get released back into the population. And they compete on this food pretty intensely. And they're exposed to natural microbes and predators and fluctuations in temperature. And then in the fall, I'm just going to show this evolving treatment. I also had a treatment where I stopped them from evolving by replacing their eggs, but I'm just going to show you in the fall, they were all evolving to fall. So they either had a history of competition or not. And so 6 generations passed in the summer and 3 generations in the fall. And I measured population sizes throughout the fall and summer. And then I phenotyped the flies at the beginning of the experiment, at the end of summer, and at the end of fall, looking at things that we thought would be important for competition and climate. So things like body size, fecundity, and development rate. And I'm going to divide the results into summer ecology, summer evolution, fall ecology, and fall evolution. So what do we find? So here is the summer ecological dynamics. So I'm showing the focal species abundance over the course of the summer season, with and without its competitor. And so we see that it does better without its competitor, which is what we expected. And its competitor is able to suppress its population growth, reduce its abundance. But what we had expected was that this would kind of lead to some kind of trait change. And we measured 10 traits in the kind of hope of capturing this. But we found no effect of this summer competitive environment on any trait that we measured, which was kind of surprising. No effect on end of summer phenotypes. And then we looked at fall ecology, and likewise found no effect of history of competition on ecological dynamics. And this is where I became slightly worried that nothing would have happened. So there's no difference in the population dynamics when they've been previously exposed to a competitor or not. But when we got to fall evolution, we found that this is where the differences came out and the effect of summer competition. So history of competition changed the trajectory of fall evolution, and it did so for 3 traits in particular: cold fecundity, female body size, and development speed. And so I'll talk about each of these. So this is body size, fecundity, and development speed at the beginning of the experiment, which is just 1 point because there's 1 population, end of summer and end of fall. Again, the dark is no competitor and the light is with a competitor. And so we see for all of these that we start somewhere, there's no difference between them at the end of the summer, and then they diverge over the course of the fall. So flies with a history of competition evolved to be larger, to have lower cold fecundity, and to have faster development. So we see a divergence in these 3 traits. So what does this mean? So this is kind of evidence that a history of competition can shape evolutionary trajectory in a changing climate, which I don't think it had been shown. And that, to me, this is a very cryptic effect of this invasive species, because the effect of competition didn't show up in the places that we most expected it. So in these fall population dynamics and in the traits at the end of the summer. And so what we kind of thought must have been happening is that something did evolve over the course of summer, but it's very difficult a priori to know which traits to measure and to know the common garden conditions under which to measure them in order to detect this evolutionary change. This is something I still struggle with, trying to detect evolutionary change using common garden traits. And so I started thinking about this and wanted to know what other studies had found. And so these are my results. The end of the summer, you see no effect of the competitive environment on any of these 3 traits. And so looking at this as an effect size, we see all overlapping 0, no effect for this competition in flies. And so I wanted to look at this more broadly with a meta-analysis, looking at competition and warming across a variety of taxa. And actually, I did predation as well, which I'm going to talk about here. And so this was a meta-analysis of evidence for rapid evolution of life's history traits, looking at what other people had found. So I was able to find 58 studies that met my criteria, which most notably, all of them had to have common gardens to isolate evolved responses. And so that really shrank the number of studies that were and the types of taxa that qualified for this. And so they either had naturally imposed selection or experimentally imposed selection. And this is what we found. So you can see that all the confidence intervals are overlapping 0. So what that means, so each point is a study And we saw no consistent effect of anything on anything. So no consistent effect of any, and this includes predation too, that I'm not showing, warming, competition, or predation on any of these life history traits across studies, which was surprising, especially for certain combinations like temperature and body size that you would think we compiled kind of the theory behind this and the predictions that we kind of think could be kind of a common outcome. And also, of the effect sizes we pulled out using our like predetermined criteria, only 30% of the effects were actually significant. So what issues does this raise? Well, first of all, I think this gave me faith in publishing negative results, because I didn't go up against any kind of backlash or questioning during review, which I was surprised about. It's just completely negative. This paper is completely negative results, and it was completely fine. So that it was nice to see, because it's, if you are looking for something and you don't find it and you try to do it well, I think it's okay to publish these kind of negative results. And then it got us thinking about when should we actually expect consistent evolutionary change. And so in the paper, we talk about the theory and the expectations behind each of these combinations. And actually, for competition and warming, they're pretty weak, and there aren't strong kind of theoretical expectations. The one area where we have more theory is with predation and body size, and that's the one response that when we teased apart what life stage they were preying upon, we actually did find one small effect. And so it's looking back at the theory and thinking about what we should expect to find. And then finally, something I already mentioned, which is this difficulty when you're trying to track evolution using traits, of choosing the traits and choosing the conditions. Because just because you don't find a change, an evolutionary change, doesn't mean there wasn't one. And so this is a difficulty I still struggle with. So the conclusions from these three studies are, the first one, warming made arrival order more important. The second one, I'd say the main take home, is that coexistence theory can and should be used to understand and incorporate priority effects. And then the final project, that past competition shaped evolutionary responses to climate. And I've mentioned kind of how I've tried to draw on these various theories in my work. I would say the first two are much more well resolved. This was very evolutionary theory light, but I try to kind of use theory as a foundation for my different studies. And so this led to a recent paper kind of thinking about the kind of how we can do this and the philosophy behind this. So this paper kind of goes through my thought process and how I've tried to use theory and how others can try to use theory in various ways when motivating empirical research. So I encourage you to take a look. And with that, I'll thank all the various labs I've been in and people that have helped me with these projects and my funding sources. And that's it.
All right, great. So happy to take questions from the audience. Maybe I'll pass the mic around and then play around with the camera here, so that actually I realized our camera, you can only see the less populated side of the room. Maybe questions from in the room? Jeff? I like waiting for students, but we'll give you one. We'll give you one.
Great talk, Tess. Thank you. Can you hear me?
Yep.
Okay. On the priority effect stuff, I'm curious your thoughts on how Lee effects fit into this story. In particular, it feels like with some of the systems we work in, with the plants at least, there may be these low density, positive frequency dependent effects, and then negative density dependence kicking in at some densities, just for amelioration of stress conditions at very low densities, perhaps. How does that Yep.
That's interesting. I haven't really done anything with Lee effects, but actually there's this recent, or not even published, paper out of my PhD advisor's lab that is, I think, the aim is to try to integrate Lee effects into modern coexistence theory. And I haven't really read it in depth yet, but it's definitely something that should be done. As you say, like if the positive frequency dependence is itself density dependent, then how does that differ from other types of positive frequency dependence? And I'm not really sure like what their conclusion was or how that could be integrated, but I know like at least one group is working on that.
Okay. Other question? I'll ask. I was thinking to the first project, and actually towards the end with your fly study, I noticed that you were measuring fecundity, which I think is totally important. And then I was thinking back with the aphid. Did you ever measure, because you're seeing all these effects of warming, but I don't know, did you look at fecundity, the effect of that, like maybe the effect of warming on fecundity, even potentially removing some of these, because that could also be.
Sorry, you cut out at the end. Removing what?
I just imagine, well, the short in the aphid, the first aphid project.
I think I missed part of that question, but so we didn't measure. So measuring fecundity for aphids is most things are easier for aphids, but measuring fecundity for aphids is harder because everything happens so quickly and you have to like remove each individual versus like a fly that lays eggs on something and you can count the eggs. So I never measured I didn't really do a lot of trait stuff with the aphids. It was all just like population level. It's easy to measure population growth. It's much harder to do anything like kind of individual with the aphids. But I imagine like, yeah, fecundity is going up with warming for them.
Yeah. I imagine it was a technical difference there. Cool. Other question, Claire?
That was really neat. A similar kind of aphid question. I noticed the species that they show at first, you mentioned that they're winter. And I was curious if you thought the would impact like when those eggs hatch and if that could like negate the effects of like those priority effects.
I think I caught most of that. Did you say could the weather affect when they emerge, or?
Yeah, does the warming affect when the aphid eggs hatch?
Oh, the warming?
Yeah.
Yeah, so then you get into like the kind of broad scale, like, so I was looking at a very like micro priority effect with like dispersal between plants and like on a very small scale, but it's hard to predict what climate change in general and what warming will do to like their relative arrival times, because the orange species could potentially get up to the north faster under warmer conditions, and they float on these like airways that could change. But then of course if it's warming, the green species is able to emerge earlier. So how that impacts their relative arrival isn't totally clear to me, but definitely like on those more macro scales, climate change probably will have some kind of impact on the arrival order between like a long distance disperser and like something that's emerging from from eggs.
Yeah. How many Zapriot if that was what there, and
I'm sorry, everything's cutting out. I can't hear. I heard Zaprionis.
We have low back microphone. Just give us a sec.
Can you hear me now?
Yes.
I was wondering like how many of the other species were in the cage with the Drosophila, and like it was very curious they didn't evolve the traits didn't shift more, and like, is there anything about how they were competing that might explain that?
Okay, so I think if I remember, we put in like 5,000 Zaprion. So we put in as many as we could possibly grow. So it was like, I think it was either 5 or 10,000, and we continued putting them in because we just wanted like the most intense competitive pressure. Like whether or not that was biologically realistic to how many there actually are, they would be faced with in nature. But we put in intense pressure on the competitive side for them. And we did see, like you could see that the Zaprionis are sitting on the food. The Drosophila melanogaster having trouble laying eggs. You could see when you go into the cages that like the abundances had reduced. So yes, we were very surprised that the that didn't exert some, even at the time I remember saying, if this doesn't exert some kind of evolution, like evolutionary response, I don't believe in evolution anymore. But like, yeah, it's just, I think it's just this, there's so many things we could have measured with the flies, you know, like of course fecundity, body size, development rate are like classic important ones to measure. But even for those, we had to choose like development rate under what temperature. Like we had to choose a temperature and measure it at that temperature. There's so many choices that went into it, and into any kind of common garden like phenotypic trait measurement, that it actually in retrospect I guess isn't that surprising. Yeah, I don't know what we could have done differently or to like detect that, or even what trait I would have measured in retrospect. There's nothing. I'm not like a fruit fly biologist, so, but I was working with fruit fly biologists, and they, you know, they also were surprised. So yeah.
Cool. Any other questions? All right. Well, if not, maybe let's thank Tess one more time. And yeah, minor tea time, 3:18 Pacific. After a few minutes to set up. So